Alrighty, welcome back to the second phase of our analysis here. So today was the day after we froze the sample, well, attempted to freeze. Apparently the addition of the unknown substance to be analyzed was sufficient to lower the freezing point of the water, and so it did not freeze after, well, like 14 hours plus. So we have to presume that there's obviously some contaminants like salt, which there was certainly plenty of after we were done dehydrating the sample, as well as some steel oxide and probably some other minor debris from the local atmosphere. We were, however, able to come up with some fairly interesting observables for you here. In the future, I have actually managed to keep the sample isolated, and it is currently in a condition where it can wait for some time before I continue with analysis and not be compromised. So I am going to be upgrading microscope at some point in time so that we can go in even deeper analysis of this visually. So let's get to it. Here's the sample after being frozen in the fridge for a little while at 4 degrees. We have some suspension, probably some dirt particles and salt particles floating in there, and a bit of a difficult to discern but very viscous sort of fluid accumulating near the top, some kind of oil, possibly from the steel, more than likely. So we will continue, and we have some sediment at the bottom as well, which is very likely dirt. And we're going to go ahead and put this in the freezer and freeze this. Initially I was going to do 2 or 3 hours, but we'll just do all night until it's frozen solid and remove it, and then we will evaporate as such. Okay. So here's our sample from last night. It still isn't frozen. If it were just regular water, it would definitely be frozen by now. Let's just have a look, see. We'll probably have a little further sedimentation. And it's a little difficult to see depending on the angle, but you can definitely see the oily substance as mentioned in the last video. So now I'm going to go ahead and start dehydrating it. After dehydration, the samples were then impregnated into resin and prepared for the slide, as you can see here. In the upper portion, you can see there's a dirty kind of yellow colored, not a molecule, but it is actually a bubble. And on the outer surface, you have a slight bluish, but mostly black and brownish colored discoloration around it. This is what I believe to be the door. Once again, the door being visible in this diagram, I believe it's door anyway. Further analysis is needed, but these findings would seem to be consistent with the entropic aspect of door, where the shape of each specific bubble isn't exactly circular, much like pore is actually very circular and very uniform in color. The experimenter would also like to note that observations at this point are essentially subjective, but as you can see in the lower part here, here's a much better defined door particle for now. And I believe that this is a pretty good example because you can see there's a sort of orange but reddish tinge to it. This would evidently be from the coloration of the salt particles of the salt bowl. However, it's interesting how they assemble into this sort of structure. As you can see, there is also some bions present in the sample, which leads to the question that is DOR really just a condition of an imbalance of DOR bions in a system with the presence of bions still only requiring some sort of check and balance system to create homeostasis for the DOR to be eliminated and subsequently for pore to reinstate itself. Once again, more samplings of bions. Although it would be very easy to interpret this cell as a bion, it is in fact merely a very well reflected salt crystal. The, of course, visually distinguishing feature being in this diagram, there's an absence of a nucleus. In the former, there was a nucleus. As in the diagram here, it's notable that there seems to be two different particles in this one instance overlapping each other. It's possible that this is the sample trying to reach homeostasis. There is obviously a bion in there by the blue glow on the one side, but on the other side, it almost looks like DOR energy. So it is almost like an image of homeostasis of the sample at some point. We'll be doing more analysis with the Radiorgonomic device just to check this out. That would be the Radiorgonomic Broadcaster / Analysis 3-dial analog box. The newly changed omnidirectional analysis dish should make this very efficient. Once again, more evidence of bions in the sample. As I'm saying, it seems more in support of the fact that every sample will have bions and pore in it. However, it is a matter of proportion of the two. And in the final photographic sample, you can see the blue streak from the previous. I just wanted to take it with the flash on so I could isolate the white from the photo so that you could visually sort of discern the blue haze left over from the bion presence. Anyway, the research will continue. It'll take some time to get a new microscope, but if you enjoyed, like and subscribe.